Yeah, it's perfect.
No, she has to use the language.
Oh,
Give me just 1 second. I'm going to explain this to her.
Hi, Dr. Osinusi, just 1 second. Can you check on the lower part of your screen where you see where it says share screen, record, reactions? The last part, it says interpretation or Spanish. It should be in English. So right next to reactions, there's the part where it should be in English. This is for the interpretation so that they know.
Yes, I've changed it to English.
Perfect. Then it should work.
Okay.
Thank you very much.
Okay. Are you seeing my slides?
yeah.
Oh, okay. Thank you. So, good afternoon, everyone. My name is Anu Osinusi, and I oversee our emerging and respiratory viruses group within the virology therapeutic area at Gilead Sciences. And part of that role includes my work as the project and development lead for Remdesevir. So first, I'd like to thank the organizers and Dr. Fernandez-Cruz for the opportunity to share the story of Remdesevir with you. And I'd like to say, this story is only possible because of the, amazing work done by thousands of researchers and scientists, chemists, healthcare workers, and most importantly, patients. But I would also like to single out the healthcare workers, the researchers, the physicians, the study staff in Spain, who really have been at the front lines of the pandemic in Spain, not only playing, taking care of patients, but also playing a critical role in all our Remdesivir trials and in ensuring that the experiences and voices of patients from Spain were heard. So moving on to the story of Remdesivir, shown here is the structure of Remdesivir. It's an adenosine nucleotide analog. The structure is shown at the top and the activity is shown in the table below. And the research that led to Remdesivir began as early as 2009 with subsequent identification through antiviral profiling in 2013 and 2014 in response to the West African Ebola epidemic. So Remdesivir itself is a truly unique and atypical antiviral agent with broad spectrum activity that is potent both in vitro and in vivo against multiple classes of viruses that are associated with significant morbidity and mortality, including coronaviruses. So just to show you, really what the journey of Remdesivir has been, between 2014 and 2019, the primary programs for Remdesivir were for acute Ebola virus disease, working alongside our partners to fully understand the safety and efficacy of Remdesivir. And this culminated in the PALM study, which showed that 2 other investigational antiviral agents were associated with greater survival. But by the time these studies were done, we then understood the PK of Remdesivir and had also generated safety data in over 500 individuals. And so can you hear me okay? I wasn't sure if you're hearing me.
Yes, I can.
Yes, we do.
Oh, okay. Thank you. Okay. So in parallel to our efforts, in Ebola, Gilead had also worked with a number of our partners to study the effect of Remdesivir against coronaviruses in vitro and in vivo preclinical animal models. However, despite positive preclinical data, we could not advance it into clinical development for SARS or MERS. as there are very limited cases of pathogenic coronavirus infections in humans to support the necessary clinical and safety efficacy testing. And then SARS-CoV-2 came along. I think the world learned about this in late December. And I actually remember getting an email from Dr. Fred Hayden, who's one of the, a renowned ID expert, from the IDSA ProMed email about, several cases of undiagnosed pneumonia in Wuhan province on December 31st, he sent that email. And then a week later, this was identified as a coronavirus. And then fast forward to where we are today with 31 million infections and over 960,000 deaths globally. And, this slide really, just shows what it was like for us also at Gilead, those early weeks, with our development program and our first patients actually being a patient dosed in the US, and fast forwarding really quickly to work with Dr. Fred Hayden, Dr. Peter Horby, Professor Bin Kao to get the China study started, the Act 1 study, the simple trials, the solidarity study started, discovery, expanded access, and just really incredible progress in a very short time frame. And so I think, what we would say is that, when the outbreak happened, we had three critical things in place. One was we had the benefit of understanding the preclinical profile of remdesivir and coronaviruses. Secondly, we had existing safety data by that point in over 500 patients, primarily treated from the Ebola trials. And then we had a scalable manufacturing process that was able to produce sufficient amounts of drug to support large clinical trials. So in parallel to the clinical activities, we were also working very closely with our partners to work on in vitro and in vivo experiments that also showed that remdesivir was effective for COVID-19. And so this is one work done by Emmy DeWitt at NIAID, one of our partners. And similar to the MERS experiment she had done earlier, this NHP study in rhesus macaques showed that in contrast to vehicle-treated animals, animals treated with remdesivir did not show signs of respiratory disease and had reduced pulmonary infiltrates from radiographs, as well as the virus titers in the bronchial alveolar lavage and lung viral loads being significantly lower with a clear reduction in damage to the lung tissue. Okay, so now moving on to our development efforts for COVID-19, I think, the common goal for us was really to determine safety and efficacy and deliver rapid broad access for the appropriate patients. And we felt the way to do that was really through three pillars. First was to establish safety and efficacy and support regulatory filings. Second was to scale up manufacturing. And third was to deliver responsible access. So moving on to the published studies, I think Dr. Lane did an amazing job earlier today highlighting some of the data from the remdesivir trials. So I think shown on these slides are published studies, three of which were adequately powered randomized trials to address two key questions we actually had back then in February. First was, is remdesivir safe and effective for COVID-19? And the second is, can we shorten treatment duration and have a shorter treatment from the 10 days that we had already used with Ebola? And I think it's also important to note that, at the time that these studies were done, there is very little known about the disease, the natural course, the progression, which endpoints were meaningful, which populations should we study. In the US, we had shortage of tests of virology kits. So people were using tests more for diagnosis and like monitoring viral load. So a whole host of questions, but I think, those were the key questions we have. The two studies to look at efficacy and safety were the randomized trial in China that I alluded to earlier and the Act 1 trial that Dr. Lane showed you some data on. And, the China study, I'm not going to go in much detail because, that study was unable to enroll the primary population and enrolled only 237 patients because of the waning epidemic, in China. So it was, underpowered, but did show a trend to shorter recovery times with Remdesivir. So moving on to the ACT-1 trial, I think, you're all familiar with this study. I think it's just important to note that the population studied was very consistent with what we know about the populations that were being hospitalized in the February, March timeframe. It was a global study that included sites in Spain, and shown below are just some key variables about the population. Most importantly, that 89% had severe disease, about 26% were on invasive ventilation, and more than half had comorbidities, which is consistent with what we've seen in other publications as well. So, I'm not showing the, this is the preliminary analysis that was published. As Dr. Lane highlighted earlier, the full data sets will be published hopefully in the next several weeks. And he'd also, shared some of the day 28 data, which is very consistent with what was shown earlier. And so, the time to recovery was 11 versus 15 days. But when you look at the subgroups, the patients that, did benefit, the most were, when it comes to recovery, were, patients with more severe disease, and really largely driven by patients, in the low flow group. And then when it comes to mortality data, I think Dr. Lane showed something that is a little bit more data than what I'm showing here. This is the day 14 mortality, where you see a trend to improvement in mortality overall population. But clearly, when you look at the low flow group, which is the majority of patients that are hospitalized, you see a hazard ratio of 0.3 with, which was statistically significant, 70% reduction in mortality in this population. And then when it comes to safety, this was a placebo-controlled study. And I think what the bottom line from this slide is really that, in both groups, safety was similar, suggesting that, the serious adverse events and safety profile you see are largely consistent with underlying COVID and driven by the underlying disease primarily. So now moving on to the next two studies. And so these were the studies that definitively helped us understand that in non-mechanically ventilated patients, 5 days of Remdesivir is sufficient, and there wasn't, additional benefit for giving longer treatment courses. And so this is the severe study, and the population enrolled here also included, sites in Spain, and 27% of the patients were on high flow or non-invasive. So this study did not, in the part A, the primary analysis population, did not include invasively mechanically ventilated patients. Those patients were then subsequently enrolled in the extension phase, which will be sharing data once those are finalized over the next several weeks. And what this study really showed was that in the, once you adjust for baseline differences, the 5 days of treatment was just as effective as 10 days in patients that are not mechanically ventilated at baseline. And then moving on to the moderate study. So the moderate study was a little bit different because this one had a control arm and standard of care. It was an open label similar to the other study, 600 patients. And once again, patients included from Spain, and the top comorbidities were cardiovascular disease and hypertension. And shown here, it's a little busy, but I think, what I wanted to just highlight here again is that when you look at the primary objective, which is to look at your distribution in the ordinal status at day 11, the primary endpoint of this study, the 5-day group had a significantly higher odds of an improved clinical status compared to standard of care with an odds ratio of 1.65. So this was recently published just a few weeks ago in JAMA. The 10-day treatment did not. However, when you then looked at, post hoc analysis at later time points, you could see that still there were higher rates of clinical improvement and lower rates of disease progression observed among patients receiving remdesivir. So I think the bottom line from both two studies were really to address the question of 5 days being just as effective as 10 days in people that are not critically ill. Okay. Then I wanted to also share this data. I think, one of, as the studies were ongoing, we thought it was also important to see beyond, the 100 or so sites you select for clinical trials, like what else is the experience, outside those sites. And so this was a contemporaneously designed study that was actually looking at patients that were enrolled during the same time frame as the severe study. Because the severe study didn't have a control arm. And so similar inclusion criteria to the severe study, but these were real world patients enrolled primarily in the US and parts of Europe and Asia as well. And so looking at recovery and mortality. And so I think, in this analysis, and we envision we'll have a more future analysis on day 28 outcomes, is that at day 14, when you compare it to this real world cohorts that had similar inclusion criteria at entry, remdesivir was, associated with, improved recovery and 62% reduced odds of death when you compare it to the standard of care cohorts. As I said, it's a retrospective study and it's real world data. So very, I think, it's important to highlight that aspect. In addition, I just wanted to also put this out there. Like, I think for us, right from the start of the development program for remdesivir, we just felt that it was very important that no patient is really left behind. And what we knew was that while there weren't as many cases in pediatric patients, we were still getting requests from pediatric investigators all over the world where there are patients or children who have more severe comorbidities and were at risk of getting COVID. In addition, we got a lot of requests in our compassionate use program from pregnant women. And so shown here are some data that were shared at the IAS conference a few weeks ago, looking at just outcomes in children and pregnant women. And as you can see, about half of those children were actually on mechanical ventilation or ECMO at baseline, and among pregnant women as well, about 40% had really severe disease, with 67% being in the ICU. And so what I'm showing in blue are the 1-point improvements. You go from IMV to high flow or low flow to no oxygen, as well as the discharge rates. And I think what is reassuring from this data is that those children and those pregnant women, it's not a controlled study, it's a compassionate use program, had significant rates of improvement or discharge by day 28. And this data would be published in the next few weeks, but it was already shared at the International AIDS Conference a few weeks back. So that leaves us to where are we right now with the next steps with remdesivir. I think really the way we see it is that we've looked at remdesivir and in this hospitalized group of patients, there's still data gaps there, looking at children, pregnant women, patients with acute kidney injury, and a whole host. Anti-inflammatory agents like steroids have been shown to really impact mortality, as Dr. Lane showed, in that critically ill, and maybe there are benefits in patients on oxygen support as well. So right now we have a number of studies that are ongoing, looking at combination therapies with remdesivir, especially in that really sick group of patients. So, we have with baricitinib, a JAK1 inhibitor, the ACT2 study, IL-6 with tocilizumab with Roche that we're doing in partnership with remdesivir, interferon beta with the ACT3 study, monoclonal antibodies with the Lilly compound, and a whole host of other targets, CCR5 inhibitors, type 2 inhibitors, anything that can really help us define is there anything else we could do outside of remdesivir and steroids to still improve outcomes in the very sick people. And then on the other end of the spectrum, we feel that it's important to still address people at higher unmet need, especially patients at high risk of getting hospitalized or having bad outcomes for COVID. So we've started 2 programs. One is to look at, like, if 5 days could be shown to work in hospitalized patients for pneumonia, is it possible to look at shortened durations of treatments in patients who are not hospitalized? And so this study is also going to be ongoing, hopefully with sites getting up and running in Spain. In addition, we also are looking at other formulations, and the one that is most further ahead right now is inhaled remdesivir, recognizing that the goal is to be able to have treatments that people can use at home that are easy to use and convenient as well for this population. So those programs are ongoing, and hopefully over the next several weeks, sites will come on in Spain, and then data will read out soon. Just a couple more slides. I think the your manufacturing, what we knew very early on in the pandemic was that, look, remdesivir is a very complicated product to make. It's very resource and time intensive. There are 70 manufacturing steps. That is a long linear synthesis process, with 20 of those steps having to be completed sequentially and several of them taking several weeks. In addition, because it's an IV drug, you need sterile manufacturing capacities, which limits the number of organizations you can actually use. So we knew we had to move very quickly, and the one thing you can't buy is time. So you can't wait for studies to be ready and then ramp up manufacturing. So what we did very early on was make decisions way back in January, way before any study started, or way before any study actually enrolled any patients, that we were going to double down and improve the, accelerate our manufacturing capacities in case remdesivir worked, and then there was, a need for it. So we instituted process improvements and refinements. We've worked very closely with other pharmaceutical and chemical manufacturing companies. I think COVID has all taught us that we need to do things a little bit differently to help us meet and exceed our production goals. In January, we had 5,000 treatment courses. By June, we had gone up 40-fold. And by the end of the year, we're hoping to target global demand, which means millions of treatment courses. I think that's just, a testament to, problem solving during, a pandemic that involves many different partners. In addition, we recognize that, there are needs in low middle income countries. And so very quickly early on, we had voluntary licensing agreements, similar to what we've done in our virology portfolio with HIV, our HIV medicines and our hepatitis C medicines, to work with 9 generic companies that would supply 127 low and middle income countries. And then finally, I think I'd like to end on our access program. I think, with our access program early on, the first patients actually were not from trials, they were from the compassionate use program because there was a dire need, and we went ahead to do that. So there are 2,000 plus patients treated through the compassionate use emergency access programs. But now that regulatory approvals have happened in multiple countries, thousands more patients have been treated through those mechanisms. So where are we today? I think today where we stand is that we've received regulatory approval for a number of countries that have helped broaden access for patients. We've generated data from clinical trials with, as of a few weeks back, over 9,000 people have been treated on clinical trials with remdesivir, and thousands more with collaborations with Operation Warp Speed and other companies to try and address what else we could do in this disease. We're also working to expand access to populations who really have not been included in trials, and identify strategies that can improve outcomes with combination strategies. And we continue to scale up our manufacturing process to meet the real-time demand while delivering responsible access. So once again, I would just like to acknowledge the work that has been done with Remdesivir right from when it was developed for Ebola to now with COVID-19. It's really been a testament to what hundreds, thousands of people can do collaboratively. I think it's a great example of what successful private and public partnerships should look like, especially when you're dealing with a pandemic. And so many partners and collaborators, including many here in Spain, have all from the start been focused on a singular goal of advancing science and tackling this hopefully once in a lifetime pandemic. So thank you for your time today, and I will stop here. Thank you.
Okay, thank you, Dr. Osinusi, for your excellent talk. I think we will have time for several questions after the talks of Professor Horby and María Henao. So if you don't mind, if you can wait until the end of these talks.
Yes.
Okay, thank you. The next speaker is Peter Horby. Dr. Horby is Professor of Emerging Infectious Diseases and Global Health. He is the former and founding of the Oxford University Clinical Research Unit in Hanoi, Vietnam. He is the leader of the Epidemic Disease Research Group from Oxford. This group is an international program of clinical and epidemiological research to prepare and respond to emerging infections that might develop into epidemics or pandemics such as Ebola, influenza, and also coronaviruses. Professor Horby is the first author of the seminal paper published in the New England Journal of Medicine about the role of dexamethasone in the clinical management of COVID. It's an extraordinary honor for us to have with us this afternoon, and his talk will be about the efficacy of dexamethasone in the recovery trial. Please, Dr. Horby.
Thank you very much. It's a pleasure to be here. My video is disabled by the host, so you can't see me.
Come on.
But I'm here. There we go. Okay, I'm here. Great. So thank you, everyone. So I'm going to talk about the recovery trial, but really with a focus on the corticosteroid dexamethasone. Next slide, please. Or can I do it myself? So it's been an evolution, the trial. So it started with early on with dexamethasone and azithromycin. Then we added hydroxychloroquine, lopinavir, ritonavir in the first level randomization, you can see here in red. And then later on, we've added a second level randomization for patients who deteriorated, so who developed hypoxia plus biomarkers of inflammation, and they could then be randomized in a factorial kind of design to tocilizumab. And so the trial's been evolving, and we've currently released results on dexamethasone, hydroxychloroquine, lopinavir, ritonavir. And we've now added convalescent plasma, and this week we'll be adding a monoclonal antibody. Next slide, please. Or can I do it? Okay, yeah, so just to say that we, what we did manage to do is start early, which made all the difference. This is the epidemic curve in the UK up to May. And if, next slide, please. And you can see that we managed to start quite early, which made all the difference, which meant that we could recruit across a large number of sites quite quickly to get good numbers into the trial. Next slide, please. So this shows you the distribution of sites. So we're active in over 170 hospitals in the UK, across the whole of the UK, and you can see the size of the green circles is the size of recruitment. So quite variable, but good recruitment across everywhere. And on the right, that shows you the number of enrollments per day over the first about 100 days. So at the peak, we were enrolling more than 400 patients per day. Next slide, please. This just shows you the first 100 days. So in 100 days, in the main randomization, we managed to randomize over 11,000 patients, nearly 12,000 patients. At the bottom there, you can see new drugs being introduced. So the light line that goes up to 770 is the tocilizumab arm. We're now over 1,000 in that arm. And the red line is the convalescent plasma arm. We're now up to, I think, about 500 in that arm. Next slide, please. So this just shows you the trial population. So overall, it's hospitalized patients with COVID. No other criteria apart from hospitalized and suspected COVID, although about 85% of patients have lab confirmed COVID. You know, we had quite a severe epidemic in the UK, so severe mortality, high mortality, about 25% overall, but very age stratified, as you'll see from the right, you know, going up to more than 40% in those over the age of 80. And the distribution of severity on the bottom right there, about 15% not requiring oxygen on admission, about 20% requiring oxygen, and about 35% requiring ventilation at the point of randomization. Next slide, please. So dexamethasone, which is what I've been asked to talk about. So we chose dexamethasone amongst all the corticosteroids. This was decided based on a couple of factors. One is a long half-life, and secondly, really the lack of mineralocorticoid potency. So we weren't running into problems with mineralocorticoid complications. So that was the reason for the choice of dexamethasone over other drugs. The dose, 6 milligrams once a day, was chosen because it's a depending on who you speak to, it's a low or moderate dose, but for ICU physicians, it's a low dose that's used in sepsis. So we felt it had good experience of being used in critically ill patients and in short courses, and so it was unlikely to cause any significant complications. Next slide, please. So this is the trial. The randomization is 2 to 1 in terms of 1 on active treatment, 2 on control. The reason for that is the controls are common across the different arms. So we needed a bigger pool of controls. You can only be included in a comparison if you were eligible for that comparison. So the controls for dexamethasone only include people who were eligible for the dexamethasone randomization, but were not randomly selected for dexamethasone. So we decided to unblind the data once we had 2,000 patients on active treatment and 4,000 on control, because we felt that gave us adequate power to see an effect. And you can see on the right-hand side the characteristics of the patients. So the average age is in the late 60s, a preponderance of males, as we saw in the UK, with a median number of days since symptom onset to randomization of about 8 or 9 days. And that's been characteristic across the trial. Patients are coming in quite late. Next slide, please. So just a bit of background on what patients got. So it was an intention to treat analysis. So we analysed patients based on what they were randomised to, not necessarily what they got. But you can see it was there was pretty good compliance. So 95% of those randomised dexamethasone got it, whereas actually 8% of those on usual care also got it, which would tend to dilute the effect in the final analysis of dexamethasone. Very few patients got any other medicines that were related, possibly treatments for COVID. So very few got lopinavir, ritonavir, hydroxychloroquine or tocilizumab, although quite a reasonable use of azithromycin, which is one of our treatment drugs, but used as standard of care for pneumonia in many hospitals in the UK, but balanced across the 2 arms. Next slide, please. So this was the overall result, positive result. So we used 28-day mortality. It was an open label trial, but the endpoint was a hard endpoint of 28-day mortality. And we saw a clear benefit of dexamethasone with reduced fatality at 28 days with a rate ratio of 0.83. But that's not the whole story. Next slide, please. What we saw in a pre-specified analysis was that the effect was clearly heterogeneous between the patient groups. So in those on invasive mechanical ventilation or oxygen at randomization, there was a clear benefit. But in those who weren't on oxygen at randomization, there was no clear benefit. In fact, the direction was towards harm, but not statistically significant. And the chi-squared was highly statistically significant, meaning that actually there's a lot of heterogeneity, and it's not really correct to pool these data. We should be presenting it stratified by these severity levels. Next slide, please. So that is the stratified result. So you can see that in the invasive mechanical ventilation group, a very big benefit, a rate ratio of 0.64, so about a sort of reduction of about a third in the mortality at day 28. In the oxygen-only group, a benefit still, but smaller, about a 20% reduction in the risk of death. But in the no oxygen group, no benefits from dexamethasone. Next slide, please. So the secondary outcomes were also in the direction of benefit. So the patients on dexamethasone were discharged from hospital earlier. So under the secondary outcomes, discharged from hospital by day 28. They had a lower risk of progression to invasive mechanical ventilation or death if they were not on invasive mechanical ventilation at randomization. So consistent effect across the secondary outcomes as well. Next slide, please. And then looking pre-specified subgroups, pretty much, the benefits were consistent across all the subgroups. So by age, clearly in the younger age groups, a bigger benefit, but you still see a trend towards benefit in the other ages. Benefit in both genders. Benefit more pronounced in those later in disease, which I think is really correlation with that that earlier graph showing no benefit in those not on oxygen. So really those two things were correlated. And I think that the most meaningful clinical interpretation is that if you don't have lung involvement with need for oxygen, then you're not going to benefit from dexamethasone. And then baseline risk, we stratified risk of death based, blinded, based on all the available data at baseline. And based on that blinded assessment of risk at baseline, you see a benefit across all the risk groups. Next slide, please. Just looking at cause-specific mortality, just because of the worry of deaths from other infections because of use of steroids, we didn't see any signal of an increased risk of other deaths from other infections in the dexamethasone treatment arm. Next slide, please. I'll just run through very quickly some post hoc analysis which we were asked to do by the European Medicines Agency. So it wasn't specified in the SAP, and we hadn't done it for the journal paper, but we were asked by the EMA to do it. So they wanted us to look at effects by different subgroups. So this just shows you stratified by severity, no oxygen, mechanical ventilation, and by age. You see the same pattern, so no benefit in the no oxygen group, benefit in the oxygen group and mechanical ventilation group, even stratified by age. Next slide. And this is, again, stratified by severity and then by no heart disease or heart disease. Again, the same pattern. You see the benefit whether you're stratified by heart disease or no heart disease if you need lung support. Next slide, please. So diabetes, again, the same pattern. Next slide, please. Lung disease, the same pattern again. Next slide, please. And kidney disease, the same pattern. So basically, the EMA wanted us to look at if there was any differential effect by your underlying comorbidities, and pretty much the answer is no. You'll benefit if you need oxygen or anything more than that, regardless of your underlying comorbidities. And actually, we've been getting more data through data linkage on the need for renal replacement therapy, and we are seeing a significant signal in reduced risk of renal replacement therapy if given dexamethasone. Next slide, please. So the summary of dexamethasone in recovery was that it reduced the risk of death by about 1/3 in patients receiving mechanical ventilation. So that's a number needed to treat about 8 patients. So treat 8 patients in ICU with dexamethasone, you'll save 1 life. And so on the right, of 100 patients treated, 12 would survive because of dexamethasone. It reduced mortality also in patients on oxygen, but didn't reduce mortality in patients who don't need respiratory support. Next slide, please. So then just to move on to a recent meta-analysis. So this was meta-analysis coordinated by WHO, by Janet Diaz and colleagues in that department, to look at all of the corticosteroid studies in critically ill patients. Next slide, please. So 7 trials were included. 1 additional trial was included after the main analysis, but didn't affect the final result. This was in critically ill patients only because most of the studies were ICU studies. So for recovery, the only patients included were those on invasive mechanical ventilation. So all the subsequent slides are just for critically ill patients. Next slide, please. So this is the headline result, actually. So the meta-analysis of the 7 trials showed a benefit that was very similar to the recovery benefit. So an odds ratio of 0.66 and statistically significant benefit favouring steroids. You can see the diamond and with above it on the favouring steroids side of the odds ratio. Next slide, please. So for an assumed mortality risk of 40% with usual care, which was based on the data in ICU patients, the use of corticosteroids reduced the risk of death to 31%. So that's 9 lives for every 100 patients treated. So a number needs to treat of 11 to save 1 life. Next slide, please. This is just showing stratification by different treatments. So this was the dexamethasone group. You can see they're dominated by the recovery trial, but also a fairly big trial, the Codex trial. And so dexamethasone, a very clear result in favour of steroids. Next slide, please. This is hydrocortisone, which was used in 3 studies, but small studies. So overall number of patients in those studies is not very big, but still on the side of benefit, although not statistically significant. So consistent with hydrocortisone also having a similar effect to dexamethasone. Next slide, please. And then methylprednisolone. There was only 1 small study, so really not possible to make any comments on methylprednisolone. Next slide, please. Sorry, can we just go back 1 slide? And I think here, if you look at the high and low doses, so the bottom one is a high dose study, and the top top 2 are high dose studies. There's certainly no evidence of a benefit of high dose. If anything, the high dose studies tend to be on the right-hand side of the lower dose studies. So the meta-analysis does not support the use of high dose steroids. Next slide. This was just looking at stratification by some other variables. So this was those who didn't require invasive mechanical ventilation at baseline, which seems to show more of a benefit in those who were critically ill but not yet on ventilation. Next slide, please. And this was looking at vasoactive medications, so sort of inotropes, etc., showing that the benefit was clearer in those who were critically ill but not yet on the vasoactive medicines. So a suggestion that it works in critically ill patients, but if you're really very critically ill, then the benefit is less clear. Excuse me. Next slide, please. This just shows the result stratified a bit further by some subgroups. So if you look at the bottom 3 subsets, so that's stratified by age, gender, and duration of symptoms, showing no difference. So same effect across those subgroups. Next slide, please. And this last slide really shows there's 1 additional study in there, the MET-COVID study, which is about 4th from bottom, which was the study that was identified after the main meta-analysis, and has been published and used high-dose methylprednisolone, just to show that it doesn't affect the overall result. Again, it's on the side of benefit, but not statistically significant, and doesn't affect the overall effect. Next slide, please. And just quickly on safety data in recovery, we did not collect detailed SAE data. We collected serious adverse reactions, and then we collected all-cause mortality. and need for renal replacement therapy, things like that. But some of the other studies did collect data on SAEs, and just to show that there's no evidence of an increased risk of SAEs in the corticosteroids arm in those studies that collected more detailed information. Next slide, please. So in summary, the meta-analysis in critically ill COVID patients, so that's those on ICU, next slide, showed that corticosteroids reduce all-cause mortality at 28 days. There's obviously a question about what happens after 28 days. Certainly in recovery, we have long-term follow-up and we have data linkage, so we will be collecting all-cause mortality going forward for up to 10 years. Next slide, please. There was no suggestion of an increased risk of serious adverse events. Next slide. The effect of hydrocortisone was consistent with that of dexamethasone, which suggests it's a class effect. It's not just dexamethasone, but it's corticosteroids in general. Next slide, please. And no evidence that high-dose corticosteroids are any more beneficial than low-dose. So overall conclusions, next slide. The corticosteroids reduce 28-day mortality in patients receiving respiratory support. Next slide. It's consistent in all examined subgroups, so there doesn't seem to be any reason to exclude any particular subsets of patients. Next slide. It's a class effect, so you can use different steroids, and that's particularly relevant for pregnant women, where dexamethasone crosses the placenta more than some other steroids, and you may wish to use other steroids in pregnant women to reduce exposure to the fetus. Next slide, please. And that we shouldn't be using high doses. Next slide. So the results, the overall results have changed practice. So the NIH changed their treatment guidelines. Well, the NHS changed their treatment guidelines the same day that we released the recovery results. NIH have changed their treatment guidelines. Following the meta-analysis, WHO released their guidance the same day on corticosteroids. And just recently, the European Medicines Agency have endorsed the use of dexamethasone in COVID-19 patients requiring oxygen. Next slide, please. So just a big thank you to the team. So a lot of people involved in this trial and also in the meta-analysis. Next slide, please. Thanks to all our partner organisations. It's a big national effort, the recovery trial across the NHS and the NIHR clinical research networks and NHS DigiTrials, because we have a lot of electronic patient data linkage, which is really useful. Next slide, please. And finally, to the the doctors and nurses in over 170 hospitals in the NHS and all the patients who agreed to participate. Thank you very much for your attention.
Okay, great, great. Thank you very much, Dr. Horby, for your excellent presentation. Well, let's move on to the next speaker, who is María Henao. It's a privilege for us to have this excellent physician with us. María Henao is a Colombian physician and epidemiologist. She is the coordinator of the Research and Development Plan for Diagnostics and Vaccines at the WHO. She has been in charge for different programs in the prevention and control of infectious diseases for 20 years in this international agency. She was a coordinator of the WHO World Diagnosis and Vaccine Research and Development Plan and participated on the eradication of poliomyelitis in Latin America and on the elimination of measles in Southeast Asia. She also directed the vaccination projects against Ebola in Guinea and Sierra Leone in Africa. Dr. Ana María Henao is the head of the Solidarity clinical trial. This global clinical trial seeks to find the most effective treatment against COVID. It's our privilege to have with us my friend, Dr. Henao, who will present us the Solidarity trial and the pivotal role of international clinical trials on COVID treatment. Please, Ana María, thank you.
Thank you. Can you hear me?
Yes.
Okay, so good afternoon. Before I start in English, I want to practice my mother tongue. So, buenos días y buenas tardes a todos. Muchísimas gracias a los colegas.
Good afternoon, everyone.
Muchísimas gracias a los colegas que me han invitado a participar con
I would
ustedes
like to
hoy.
thank you for the invitation to
Y
be here with you today.
desafortunadamente voy a tener que hablar en inglés.
Unfortunately, I will have to make my presentation in English, though.
Okay. So now move on. So what I am going to present today is an international effort. And I think in the context of this pandemic, as Dr. Tedros says, national processes are not what will find the solution, but solidarity, global solidarity. So I work in a team that is called the Research and Development Blueprint. This is a team that was constituted in WHO in 2015 in the context of the Ebola pandemic, with the recognition that isolated individual efforts are not the best in the context of pandemics. And we are doing this on behalf of the 194 member states of WHO. So WHO has worked since the beginning in vaccines, diagnostics, and therapeutics since the 10th of March when we activated all our processes. This slide just summarizes the work we have done in preclinical, phase 1, phase 2, and phase 3 for therapeutics. In terms of animal models, I am very happy to announce that a review of the work that has been done with our almost 100 collaborators all around the world on animal models to evaluate vaccines and therapeutics will be published this week in Nature. So you will all have the opportunity to review that. We are also in the process of summarizing all the work on the assays. We are going to talk today about the work we have done on clinical trials, our target product profiles, that is the public health perspective of the attributes that the drugs should have to be used in the context of public health, is on the web. And now we are working on prioritization of therapeutics, ongoing work since the beginning of the epidemic. What we are trying to do, we are very happy in this pandemic because many people understood that it's important to generate randomized evidence. But as we always say, people understood the word randomization, but omitted the word large. And as you can see, there are nearly 2,000 clinical trials registered in the clinical trial registries, but the majority of them are small, have only a few hundred patients. Some even never recruited any patient at all. And that's a shame because even though between the Solidarity and the recovery trial we have, and with the trials at the NIH, we will have maybe 25,000 patients. If you consider the number of hospitalized patients in this pandemic, we have only a small fraction of those patients that are occurring. So because of that, we decided in WHO under the R&D blueprint that it was important to create a multinational effort, an effort that will go across the globe and will not cover only one nation. And then we asked ourselves what are the critical questions for public health. How can we really contribute and not do just to be an also ran? And it was very clear for us that all-cause mortality was important because what makes COVID disease so terrible, that what makes us be afraid of this disease is the mortality rates. So all-cause mortality was critical. We also felt that our duration of hospital stay and need for ventilation or intensive care was important, especially for countries where the health system will be challenged by the overwhelming number of patients. So this is how the objectives of the trial were decided on the basis of the critical questions for public health. Then the second point that we discussed in the protocol, when the protocol was first designed in early March, was how can we simplify the procedures in order to allow very busy medical doctors like Vicente, who was one of our first doctors in the trial, participate and be able to contribute to a randomized trial without this becoming a burden for the work. So this is what is the trial with no papers. Everything is done electronically. The patient enrollment, the randomization, and also the follow-up. Also, the process takes only a few minutes, and the randomization information is presented on the screen and also emailed to the person conducting the randomization. The study population, this is a study about adults who are in hospital, hospitalized adults that have a definite COVID diagnosis and that are willing and consented to participate in the trial. We obtain only the information that will be required for the analysis. So this is a protocol in which every variable that is collected is because it has a purpose for the analysis. So we collect information on the country and the hospital, We also obtain documentation that the patient has consented to participate in the trial. We collect information on automatic identifiers for the trial, and also we get basic characteristics of the patient, whether or not he has any of the known underlying conditions that could aggravate the course of the disease. We also document the situation of the patient at the time of enrollment. So we want to know what is the severity of the disease at the time of randomization with very simple approaches. Is the patient short of breath? Is the patient already receiving oxygen? Or is the patient already receiving ventilation? And then if there is an X-ray available, this is not required, but if it's available, we document what kind of abnormalities are observed in the X-ray. When we started with 5 study arms. So the patients can be randomized to the local standard of care, any standard of care, or to 4 drugs, Remdesevir, hydroxychloroquine, lopinavir-ritonavir, or lopinavir-ritonavir plus interferon. At the discharge, when the patient leaves the hospital because he or she unfortunately died or is discharged, we collect basic information on which study drugs were given, whether or not mechanical ventilation or intensive care was given, and the date of the discharge and the cause of the death if the patient died. The sample size was not specified. We recognized from the beginning that the larger the number of patients included in the study, the more robust and the more confidence we will have in the results. In terms of add-on studies, the study is flexible. There is a core protocol, but also there is the possibility for different study groups or researchers to add additional observations. In fact, some of the studies that are participating in the Solidarity trial have added specialized measurements. For example, the Discovery trial, that is a sister trial of the Solidarity trial, or the trial in the Scandinavian countries, or in India. So we have this flexibility. So it is a global trial with a core protocol, but the possibility to add additional measurements. It is an adapted design, so we have been reviewing with the support of our independent Global Data Safety Monitoring Committee, the accumulated evidence, and thanks to their advice, we have, for example, as it was announced in July, stopped the randomization with hydroxychloroquine and also with lopinavir-ritonavir. Moving forward, we are going to analyze, and this is going to be available very shortly, thanks to all the investigators and their teams. We are now finalizing the process of data revision and cleaning. And so we are in the process of conducting the first interim analysis that will be made public shortly. We have an independent Global Safety Monitoring Committee. We also have all the investigators that meet regularly, and we have an executive group among the selection of the investigators who act as a steering group for the trial. In terms of quality, because I insist on this because some people say, oh, this simple trial maybe is not of good quality. We have a tool, an electronic platform that allows a global data monitoring. In addition, the teams have organized global monitors, and we have an independent process for data cleaning and verification. These are the countries who are in the trial. So I am very proud to stand here on behalf of 450 hospitals and their medical teams. 28 member states of WHO are already enrolling patients, and 14 are ready to start next week. And we have represented all of the 6 regions of WHO. This is very important because then the results will provide this geographic diversity and this diversity in the countries. The other thing that is important about having this diversity is that despite the disease incidence has varied among the different participating countries, it has no effect on our ability to recruit patients. We are recruiting about 2,000 plus patients per month, and this is likely to increase with the additional countries join, despite the variations in the transmission and the disease in different countries. So a national trial will be more sensitive to the local transmission rates. Such an international platform is free from that because while the disease declined in Spain, unfortunately was increasing, for example, in Peru. So the recruitment rates were maintained without any negative effect. So what is next for the Solidarity trial? We want to finalize the interim analysis and the publication. And I just want to say that, for example, in terms of Remdesevir that was presented today, we have about 2,200 patients recruited. In terms of interferon beta-1a, we are close to 2,000. And we have more than 1,000 for hydroxychloroquine and for lopinavir, ritonavir. When I say the numbers, I mean the ones receiving active drug, excluding the controls. So, and that was the aim of the trial, to do a trial where you have robust numbers that actually include or double the existing evidence. So this, and I'm going to finish with vaccines, have given us impetus with all our partners around the world to do exactly the same for vaccines. As you know, we have nearly 200 vaccines out there, 35 are in clinical phase, and we are very pleased that 9 are already in phase 3 trials. But we know that the world needs more than one vaccine and that the rate of attrition of vaccines and their development is very high. This is why we want to give equal possibility to all the candidate vaccines to be tested using robust trials. So we are already launching, and Dr. Tedros announced to all the ministers of health of WHO last week that we are going to do a similar trial for vaccines. And we already have about 15 member states of WHO who will sign for this trial. And we hope that this is a trial that will empower the countries and the researchers in the different countries, that will promote international collaboration, and that will eliminate the inefficiencies of conducting separate individual trials. This just appeared in Nature, and I think this summarized this talk. We think that it was good, that many people understood the need for randomized evidence, but as I said, the part of large randomized trials was missing, that the recovery and the solidarity trials with more than nearly 25,000 patients recruited among the two of them have set new standards and have shown that the combination of old-fashioned randomization using established clinical trial networks and capacities in the countries, facilitated by modern technology, using only electronic platforms, will make the magical randomization possible. And the magical randomization will lead us to have robust estimates and information that can be acted on, as Peter mentioned regarding the dexamethasone. And as I say, we are very confident that our results, and thanks to Vicente and all the colleagues in Spain that were among the first researchers during the Solidarity trial, will be able to contribute to the discussion and the data on how to best treat patients with COVID. Thank you very much.
Okay, thank you very much, Ana Maria, for your excellent talk. Thank you. And I think Professor Fernandez-Cruz can give us some questions that we have from the audience.
I enjoy immensely the presentations, and I think this is a very interesting and important roundtable. So we're going to go for burning questions for Osinusi. And Pere Domingo start by questioning, since the most damaged organ is the lung, why did you not choose hard lung endpoints, such as intubation, or respiratory failure related death in your Remdesivir trials? And there is another question very much related to this, which is from Vicente Soriano, who said, you mentioned plans for moving to Remdesivir inhale as treatment. About aerosols for pre or post-exposure prophylaxis, any plans for long-acting formulations? I know anytime you want to answer these questions, it will be okay for us.
Okay, thank you. Can you hear me okay?
We do.
Oh, okay, thank you. So the first question, I think that if I understand correctly, was because, COVID-19 is a respiratory disease, why were, endpoints such as, intubation or such endpoints not picked? I think it's important to note when these studies were designed. So they were designed in early February. And, at that point, we knew very little about this new viral disease. And so I think, the folks who are really at the forefront, who had also done a lot of influenza work, an ID specialist and, Dr. Horby here, as well. At that time, it wasn't really clear what the best endpoints should be, and no one even really knew what the mortality rates were. So I think, what came out of that was, this use of the ordinal scale, which was used in the NIAID trial, the initial China trials, based on more of like an influenza model. But part of what that captures, it was really to come up with what's the best way you can look at progression and also look at recovery, right? And so you're looking at people discharging, but also on the ordinal scale, you're able to look at different categories of the ordinal scale and what they mean. And so those are all additional endpoints, such as, progression to mechanical ventilation, days on oxygen support, and such endpoints are actually, my understanding is that, the Act 1 trial, for instance, is possibly going to be including those type of other endpoints in the trials. So I think it's a really good point that, going forward, we've learned a lot more and we know much more now in February than we did in February. And then I think the second point around inhaled remdesivir, as you point out, I think, there have been in healthcare settings in particular that, the use of, nebulized products could aerosolize some virus. So the way we think about, inhaled remdesivir and the device selection is going to be critical, using devices that have filters and, really trying to position that for people using this at home. I think some of the data that has come out of the CDC and the NIH around the risk of aerosolization has shown that it looks like it's more of a theoretical risk, but it is something that we definitely are going to be evaluating in our studies going forward. And we recognize that, inhaled, we really want to come up with a formulation where people are not necessarily going into the doctor's office, because I think we've heard that people don't want you to keep coming back to their office if you have COVID because of risk and spread for other patients. So we need to come up with formulations that are going to be able to be used outside of traditional healthcare settings. So, the first of it is inhaled, but we're also looking at other formulations. At this point, we don't have an oral formulation because, remdesivir, it's not bioavailable. Bioavailability is really low, but we're looking at other ways to address that, subcutaneous formulations, and also looking at other targets as well. And then the third thing was about PrEP, I think, post-exposure prophylaxis. I think those are all interesting concepts. I think what we wanted to first start out was, does remdesivir add any value? Does it even work? And I think, from the Act 1, and also addressing people who are the sickest. So all these trials that, Dr. Estrada and Professor Horby talked about are in that sick group. But I think now the next wave of therapeutics, the monoclonal antibodies, the antiviral agents from Merck, are looking at the outpatient, and then hopefully this will expand to, the pre-exposure and maybe post-exposure setting. And so that's the way we're thinking about it as well. And, we're happy to, talk with a lot of investigators who have been thinking about these issues. And I have to shout out to Dr. Soriano, who was, critical in all the remdesivir trials as well here in Spain.
Brilliant. Thank you so much. Vicente, you have a question for Peter, right?
Can I
Yes.
add to that question?
Go
I
ahead.
just want to clarify on the question of mortality. Once we publish our interim results of Solidarity, it will represent that 75% of all the evidence available in the world regarding Remdesivir. And we are measuring mortality, duration of hospital stay, and need for intubation. So when we publish, because I just say we have 2,200 plus randomized to Remdesivir, that will represent 75% of all the global evidence put together. And I think we will have the information that was requested.
Terrific. Okay.
Yes.
Vicente, go ahead.
Oh, no, I was going to say thank you. And I just wanted to say, thank you to the, the WHO team. And I think, as Dr. Ana Maria has said, with pandemics, ways to look at innovative ways of doing new trials, and large global trials are extremely important. And I think all the different partners we've worked with, even with the smaller trials, I think have all contributed incrementally to patients all the way back from February and even now and ongoing. So I think it's a really good way to think of future private-public partnerships and doing things different ways with pandemics in particular.
Terrific. Okay. Vicente.
There is a question for Dr. Horby. Yes, Dr. Horby, are you worried about the viral excretion? Steroids can prolong viral shedding, and this might be harmful for some patients. Is there any population that might be more vulnerable? Is this really a real concern?
We don't hear you.
Sorry, muted. Yeah, I mean, we didn't measure viral loads. I think it's difficult because I think even as you saw in some of the Remdesivir studies, it's not necessarily a good measure, particularly upper respiratory tract PCR positivity is not necessarily a good measure of infectivity. It is not necessarily a good measure of what's happening in the lower respiratory tract. So we didn't measure it. I mean, in the past with MERS and SARS, there's been some suggestion with higher dose steroids that you can get prolonged viral replication. But I don't think in practice it's been shown to be a problem. And I think in our study, we didn't see any signals of any problems. I think there might be a theoretical concern of, infectivity, etc. But, given the amount of SARS-CoV-2 viruses there is in the world, and the fact that people are being discharged all the time who are PCR positive, I don't think it's a significant concern, really.
Okay, thank you so much, Ana Maria. Great job. Great job you did from the RMSA to work on all those trials. So now we move on to the next table. So Jessica, it's your turn.